Hi, I am Bezawit, and in this video we will explore the mechanisms of overproduction and selective pruning during development. Once axons reach their target tissue, they start forming connections. During early development, multiple axons innervate the same tissue by forming redundant connections. Over time, precise neuronal connections are formed by eliminating the unnecessary or weak connections and selectively strengthening the functionally important connection. The transition from overproduction to selective pruning is mediated by activity-independent and activity-dependent processes. In the activity-independent process, synapse formation is based on the presence of attractive molecular cues or the absence of repulsive cues. Growth factors are examples of attractive cues. There are various types of growth factors that facilitate the proliferation and survival of neurons that have the required receptors. Let's look at BDNF, or brain-derived neurotrophic factor, for example. In one experiment, researchers expressed red fluorescent protein in pyramidal neurons that secrete BDNF and studied whether these neurons affect the proliferation of nearby neurons labeled in green. We can see that the presence of BDNF facilitates proper growth of dendrites with complex arborization. In comparison, acceptor neurons that are in the vicinity of a BDNF deficient donor neuron have abnormal arborization. This indicates that BDNF is essential for proper dendritic branching and could facilitate the proliferation of neurons that have the TrkB receptor. The second mechanism of pruning involves activity-dependent processes. One example of activity-dependent synapse formation is the synapse formed between Purkinje cells of the cerebellum and climbing fibers originating from the inferior olivary nucleus of the superior medulla. The synapse formation proceeds in 4 steps. Initially, at postnatal day 3, functional synapses are formed by multiple climbing fibers innervating the soma of a Purkinje cell. At this stage, these fibers are able to release glutamate and evoke excitatory postsynaptic currents that have similar amplitudes. Between postnatal day 3 and 7, functional differentiation of synapses occurs due to competition between climbing fibers. Some climbing fibers start releasing more synaptic vesicles and elicit EPSCs of higher amplitudes. Subsequently, the climbing fiber that evokes the largest EPSC becomes selectively strengthened. The molecular mechanisms underlying the selective strengthening of a single climbing fiber are not fully known. It is known that Cav2.1, a PQ-type voltage-gated calcium channel located at presynaptic terminals, is necessary for the biased strengthening of a CF. Compared to wild-type mice that attain the strongest synapse at P7, Cav2.1 knockout mice have multiple strong synapses by the end of P7. However, Cav2.1 is not sufficient for the subsequent selection of a single CF after P7. One hypothesis is that climbing fibers may also be competing for a limited resource provided by Purkinje cells, and that the amount of this resource increases throughout development. The third stage of synapse formation involves the translocation of the strongest climbing fiber from the soma to the dendrites. The weak climbing fibers, which innervate the soma, are eliminated in 2 stages. Let's look at some of the mechanisms underlying the early phase of CF elimination, which occurs between postnatal day 7 and 11. First, the activity of the winner CF is necessary to drive calcium influx into the Purkinje cell and further strengthen the synapse. Insulin growth factor 1 might also be involved in enhancing the strength and survival of the winning climbing fiber. Besides climbing fibers, inhibitory cerebellar basket cells also innervate the Purkinje cells. Increased GABAergic input from basket cells results in the relative weakening of the losing climbing fibers. In summary, the winner fiber is strengthened by potentiation in molecules such as IGF-1, while the losing CF is weakened due to synaptic inhibition. The late phase of CF elimination occurs between postnatal day 12 and 17. In the first mechanism, activation of a metabotrophic glutamate receptor signaling stimulates the expression of semaphorin 7A, which can bind to plexin C1 on the weak fiber to favor its elimination. In the second mechanism, the Purkinje cell releases BDNF, which must bind to TrkB receptor. However, throughout development, the expression of TrkB on weak fibers may decrease, which causes lack of trophic support. Removal of weak synapses is also facilitated by further strengthening the winner CF. For example, the Purkinje cell can release semaphorin 3, which stabilizes and enables maturation of the winning fiber. Other cell types are also involved in this process. For example, cerebellar astrocytes, known as Bergman glia, can wrap around the winning climbing fiber and strengthen it. To summarize, activity-dependent synapse formation in the cerebellar cortex is initiated by multiple innervation of Purkinje cells by climbing fibers, followed by competition among fibers. Eventually, the strongest fiber is selected to innervate dendrites, while the weak fibers are eliminated by Purkinje cell autonomous and non-autonomous mechanisms. The final outcome is monoinnervation, in which a Purkinje cell is innervated by a single climbing fiber. In this video, we have learned that axons form multiple connections once they reach their target tissue. Connections that are functionally important for proper development of the nervous system are kept, while non-essential connections are pruned. And this transition from overproduction to selective pruning is mediated by activity-independent processes such as secretion of growth factors and spontaneous neural activity-dependent processes such as competition between